herbicide resistance is such a challenge for weed management, but having herbicide tolerant traits in crops like, corn or sorghum or cotton or soybean is such a beneficial thing for growers. It helps them to have a very good weed management and then also rotate herbicides and whatnot.
Welcome to the Crop Science Podcast. I'm your host, Brian Ernell. Today I'm excited to have Dr. Mithila Jugulam from Kansas State University with us. Her research program focuses in weed physiology in the agronomy department at Kansas State, and it encompasses weed problems in crop and rangeland systems with an emphasis on herbicide mode of action and fate and herbicide plants in the environment. Specifically, the research is focused in the area of herbicide resistance, including mechanisms and inheritance of herbicide resistance in weeds, the effect of elevated temperature on herbicide efficacy, and the identification of sources of herbicide tolerance in crops. Well, Mithila, welcome to the Crop Science Podcast.
Thank you very much, Brian. Thanks for having me today.
Can you tell our audience a little bit about how you ended up at Kansas from where you came from and what you do at K-State?
Sure. Thank you again. I'm originally from India, where I obtained my BSc and MSc in agriculture in two top land grant institutions. I'm pretty much aware of how the research and education in land grant institutions usually work. And after my master's degree, I also worked with the Indian government as a scientist with Indian Council of Agriculture Research, which is pretty similar to USDA in here, US. After working for a few years, then I moved along with my husband to Canada when he got a job opportunity. At that time, I wanted to pursue further education and applied for a PhD program, yet another big land grant institution in Canada, which is University of Guelph. I'm fortunate to get accepted in weed science program there. That's how my weed science exposure, especially to weed physiology and molecular biology, I did my PhD work in those areas. And after working as a postdoctoral fellow at the same university in Canada, I was hired as a faculty here at Kansas State University in the same area of my expertise, which is weed physiology. So I've been with Kansas State since 2011, October. It's been a long time now, but I really enjoy my work here. So that kind of gives you a background about how I ended up here at K-State. But in terms of what I do at K-State is I have both research and teaching appointment, but I have 70% research and 30% teaching appointment. So like you described in the introduction, Brian, I have multiple areas of research related to weed and herbicide interactions. In terms of looking at a big picture, majority of my research is focused on why we have some weeds evolving resistance to herbicides while we can control other weeds very easily. So we dig into deep, look at molecular level, physiological level, as well as, we do a lot of whole plant analysis as well. So that kind of is about 60% or even close to 70% of my research. And as you mentioned, we also do quite a bit of work looking at what are these environmental factors that can affect our weed performance, or when you apply herbicides, whether weeds can be killed or not. Not necessarily it is related to resistance. Sometimes environment can affect the herbicide performance, thereby challenges with weed control. In this regard, we have looked at temperature, in particular, how increased temperature can affect weed management. We have looked at multiple weed species that are important here in Kansas and across US as well. And more recently, we have invested quite a bit of time in looking for herbicide tolerant traits in crops. While herbicide resistance is such a challenge for weed management, but having herbicide tolerant traits in crops like, corn or sorghum or cotton or soybean is such a beneficial thing for growers. It helps them to have a very good weed management and then also rotate herbicides and whatnot. So this is also, I believe, is a very important area, especially in minor crops where industry is not going to invest so much. As an academic institution, if we invest our time looking at what are our opportunities in improving crops like sorghum, for example, in terms of herbicide tolerance, how can we get benefited from them in terms of weed management?
Explore the future of agriculture with KWS, a global leader in innovation and sustainable farming practices. Uncover the exceptional qualities of our hybrid rye, cultivating a legacy for a greener tomorrow. Visit kws.com/US for more information and for dealer locations. KWS, seeding the future since 1856.
So that's interesting. I also work in a lot of the minor crops, sorghum specifically. What kind of work are you doing in sorghum as far as looking for crop tolerance, and how do you have to juggle the ability of sorghum to cross with some of their weed species?
Yeah, excellent question. That's something we always think about before even starting this kind of research, right? Ultimately, all the effort we put in identifying the tolerant, in the end, if it's not going to be helpful, that's not going to work. First, to answer your first question, as probably you and many of your listeners know, we do have some herbicide-resistant sorghum technology available now, like double team or InZen sorghum. They are there for growers. So we are focusing on herbicides that are not already being investigated and available for growers. In that regard, we have spent quite a bit of time in looking for tolerance to HPPD inhibitors, which is one group of herbicide. Usually they are referred as bleaching herbicide. They are widely used in corn because corn is naturally tolerant to those herbicides, but not registered for use in sorghum. So we started to look for natural tolerance in sorghum. We have screened a large number of genotypes collected from across the US and around the globe as well. In that regard, we were able to identify a few genotypes that showed tolerance. And as a next step, we also looked at why are these tolerant? What makes them to be tolerant? In terms of mechanistic studies, we have done quite a bit. And more recently, we are also looking for tolerance to PPO inhibitor herbicides as well. And another group like ACCA inhibitors like clothidim or fusarofop. Those are the other targeted herbicides we are looking at.
So while you're screening for tolerance in sorghum, are you also screening for tolerance in Johnson grass to see if you could bring a gene across like they would, say, in weedy rice moving into cultivated rice?
Yeah, that's a great question, Brian. In fact, if you look at how the InZen sorghum was developed, is the gene came from the wild species, right? They identified resistance to ALS inhibitors in wild, and they brought it the university, and they made crosses. Yeah, it's the same thing. Yeah, we have done some screenings of Johnson grass as well as sugarcane collection, but we couldn't find any tolerance to HPPD inhibitors in those. Obviously, only two weeds so far, luckily, only two weeds in the US that are resistant to HPPD inhibitors like palmer amaranth and water hemp. Palmer amaranth is a huge problem in sorghum production. So yeah, that's there. but luckily not in our, we don't have it in wild species related to sorghum yet. So to answer your question about, all right, if we are successful in getting the HPPD inhibitor trait in grower's field, whenever it happens, what if it moves into the wild relatives, right? Two ways I look at it, Brian. There was some good work done at Texas A&M looking at what's the gene flow kind of from sorghum to wild relatives, and what would be the progeny in terms of their fertility or germination in the field in nature. Based on my understanding, it's not going to be so big of a problem in terms of how many fertile progeny we get, or even they germinate or not. That is not uncommon if you look at wheat versus jointed goat grass, or even canola and related wild mustard species. When they moves to wild, essentially because of difference in species, there could be an issue in terms of fertility of the offspring. So that may help us in management, but we cannot take that for granted. Mother Nature is very, very, unbelievable. You may end up having more progeny. So my answer to that is, luckily, the genotypes we identified that are resistant to one herbicide within the HPPD inhibitors, like Callisto or mesotrione, they are not the same that we identified for Ladus or Tembotrione. So, which is good in terms of management, assuming that our sorghum that is resistant to mesotrione is moved to sugarcane or Johnson grass, at least Tembotrione can work to control them. So that helped us in terms of not identifying the same genotypes resistant to all HPPD inhibitors. This kind of difference really helps us for the management if or when it moves into the wild species.
And that's one thing I try to remind farmers when they speak of this transition of genes across the wilds, is that if you're following the best management practice of the herbicides that we're utilizing, that practice takes into account any potential crosses, because you're trading herbicide modes of action, and therefore any progeny that does have survivability should be included and gotten rid of. Which is also kind of leads into the next question. With a crop like sorghum, and you could transfer that to wheat and others that have near relatives, would we ever see like a triple or quadruple stack like we might see in a cotton or in corn?
I see, again, talking to some of the growers, they like it. They like to have stacked resistances. So one good thing about obviously the need for it is very good, very high. I would say, even if it is done in academic institutions with publicly available material, we should be able to cross them and identify a progeny having resistance to two herbicides. In my opinion, that is very good in terms of management too. That gives the option for tank mixes of herbicides, and probably help us to manage herbicide resistant weeds as well. So you are not depending on only one mode of action. While stacking, you obviously look for different modes of action of herbicide groups.
So as we move, kind of moving into the next topic, you mentioned the effective temperature. Can you kind of fill us more in on some of the work that you've done in that realm? Because that was a very interesting comment that I took a note of and I want to dive into. So
sure, yeah.
Start with that.
So this is all started again with my collaboration with extension folks. You probably know our previous extension specialist, Dr. Curtis Thompson. He did a lot of work with sorghum weed management. So I just joined here, I think in 2013 or so. I was talking to Dr. Thompson. He said, "Oh, I see, this is an observation, Mithila. I see when I apply herbicides early in the morning versus when I go and apply, depending on the wind and other factors, you cannot go and apply whenever you want. There are some things to consider. I see a big difference in terms of weed control. Uh, Obviously, he understands. I also understand that that doesn't mean there is resistance in the weed. For resistance, we have to do a thorough analysis. But this, if this in the same field, some plants are controlled at when you apply one time of the day versus later when you see temperatures are rising. So then I thought, okay, this is something that we can investigate and see what exactly is going on. What we did was first we started with palmer amaranth plants that are known to be susceptible. Again, we took the HPPD inhibitor as an example. And we have access or facility here at Kansas State University in our agronomy department. They're very well controlled growth chamber facilities. So we grew palmer amaranth plants uh, under two different temperature regimes, exposing them to high temperature, some at optimum temperature, some at even low temperature. Then we grew them, and at right stage of their growth, we applied mesotrione, starting with very low dose to high dose. We did dose response studies and all. What we found was when these plants were exposed to high temperature stress, they are not able to be controlled well versus when they are at low temperature. It was done multiple times and we saw the same result. Again, we went, as a physiologist, my next interest is why is it? What is happening to plants at high temperature? Why are we not able to control them when they are grown at high temperature? So when we did the physiological analysis, we found that the metabolism or metabolic ability of plants is very high at high temperature. The herbicide is getting degraded much quicker, therefore plant is able to survive. So that kind of the first work in this area we started. Following that, we have also done a lot of work with palmer amaranth, which is resistant to 2,4-D, water hemp, and kochia. Kochia is a big problem for us on the western side, and we also have done some ragweed work in collaboration with Dr. Amit Jala in Nebraska. So these are few weed species we worked with.
So when we talk about high temperature, can you classify what in your study was considered high?
Yeah, it's a very good question. In fact, a high temperature we pick for sorghum is not going to work for wheat or soybean, right? How we pick the temperatures is that, so we take the weather data from our weather data library and look at the average temperatures during sorghum growing period, and at the time we apply the herbicide, like post emergence. When do we plant? Late in May, early June, and then when will the plants ready? So we take the temperatures from the database for several years and see the average temperature, and we pick that high and low temperature to set up our growth chambers, and then we pick low temperatures like 10 degrees below that and high temperature a bit 10 degrees above. Just get an idea of what would be the...
Did you maintain constant relative humidity, or did that change with temperature?
Yeah, that's a very good question. See, in growth chambers, when you do these experiments under controlled growth chamber conditions, if we are looking at one factor, in this case, the temperature, we cannot manipulate other conditions, right? It has to be same across. So in that regard, our other like light intensity or relative humidity, they are all kept constant. Only the variable we looked at was the temperature.
So if you were to look at it, because often especially in sorghum, as we increase temperature, not always, but often you'd probably expect a decrease in humidity, at least in Oklahoma, Texas, and Kansas. As we get hot, we get dry. So do you have any research that looked at the humidity, or can you postulate about how the humidity would also impact the results?
Yeah, no, we really thought about that, and I agree with you that in the real life or in the field, the humidity and also moisture are not necessarily always be the same. So, to answer your question about humidity, yeah, humidity is an important component. But if you look at different herbicide groups, the the effect of humidity on herbicide absorption or uptake is more prominent to some herbicide like glyphosate. Definitely, we have seen pretty clear. But on the other hand, some of the systemic herbicides like HPPD inhibitors or 2,4-D auxin, synthetic auxin herbicide, rather than humidity, it's all about the temperature. Humidity may influence. I'm not ruling out that factor, but I do not see the results may be drastically different if we had done this in the field conditions.
That's interesting, and I was wondering about just the plant metabolism. Probably temperatures of greater influence, unless there's drought stress. So, have you looked at this, or just thinking physiologically, how does drought stress influence the metabolism of a plant?
Yeah, yeah. One thing we have looked at in terms of drought stress, again in collaboration with Dr. Jala over in Nebraska, that those experiments were done over there. They had a night system where they can maintain how much moisture or how much water has been added in each pot each day. So, they measure the volume and they come up with how much. So, in terms of that, definitely there was a big effect in terms of moisture stress and weed control, which is given. But you asked about metabolism. Such work we have not done, and I don't think in the literature, if I remember, anything was looked at into weed management. I mean, in crops, a lot of work was done again. In weeds, the the main requirement to study such moisture stress effect and herbicide efficacy is we need to have a very good controlled moisture supply system in the greenhouse. So we haven't done. I don't know exactly how it can work. If I want to speculate, definitely the metabolism. When plants are under stress, the growth is obviously affected. But as a defense mechanism, probably they work these enzymes that are known to metabolize, may be much more active in working on those herbicide molecules.
That's fascinating. Any other research you'd like to highlight?
Yeah, I probably would like to, which is very exciting as a scientist to me, and I see in the future some potential also. So I mentioned one of the expertise of mine is looking at the molecular mechanisms. So when we looked at glyphosate resistance in palm amaranth, which is widespread across Midwest or across the US, the mechanism ended up being very similar to what you see in microorganisms. Like, you have this plasmid kind of vector existing in the microorganism cell that also carries some genes. So what we found in palmer amaranth is something similar. In the palmer amaranth cell, plant cell, the genome or the inside the nucleus, we also have seen such structures like plasmid, which carried glyphosate target gene. It duplicated on its own because it has autonomous replicating units. It doesn't follow any Mendelian rules at all. It's on its own, just duplicated up multiple times. And we have seen plants with more than 100 copies. That means they can make so much enzyme. Even if you apply 10 times the field recommended dose of glyphosate, you can't kill them. So that was very exciting to know. First time in the plants, that kind of discovery, we were able to publish it in PNAS journal. That was pretty exciting. I would say the eureka moment in my career.
It's absolutely exciting from a weed scientist, but from somebody trying to kill weeds, it's scary.
Scary. No, I completely agree with you. And we thought about it. Oh, how can we stop these plants making? Is there any way we can do? So we looked in the literature. The mechanism I just described is very common in cancer cells in humans. So humans that have these cancerous cells, they tend to just can't respond to any medication because of this kind of. So what they found was in the lab, if you have such kind of duplication going on, if you take the selection from that particular molecule or the chemical, these these structures, circular structures, they just dissipate. They are not going to be as active when you have the selection going on all the time. Based on that, what we hypothesize is, for example, grow the glyphosate resistant plants in the greenhouse without just applying anything, any herbicide or specifically glyphosate on it. Get next generation seed produce. See couple of generations. See if the number of copies are going down. Eventually, if we can make them susceptible. That's a long term. Obviously, in the field, it's not going to be easy to do. But as a proof of concept, we are doing that work in the greenhouse condition, growing the plants, not exposing to glyphosate, multiple generations, see if these plasmid like structures can disappear. Yeah, it's a challenge.
So so as you kind of talked to that, what's now the future of that? I mean, what's the possibility of stopping that process? I mean, is you mentioned you're looking into, but do you have high hope, or is this something that we have to attack with another chemistry?
Oh, definitely for a immediate approach, we have to apply other chemistry. I know if you look at glyphosate resistance in palm amaranth, the same population already are resistant to atrazine. I mean, that is very common in pigweeds, correct? So usually those multiple resistant populations are very common, but not. Having said that, I talked to extension folks. Probably you know Brian very well. Maybe glufosinate may work well in controlling the glyphosate resistant palm amaranth. We do have a couple of glufosinate resistant population. I'm not saying we don't, but it's not widespread. Or tank mixes and see how the plant respond. Those are immediate things to follow. Now, on the other hand, we are also looking at a new technology called RNAi.
Mm-hmm. I was wondering if you're going to bring that up.
Yeah, it's common in other pest management systems like pathology. It's very well tested, and also entomology folks have good data on that, how they can control the insect or plant pathogens using this technology. In weed science, it's relatively new, or very few labs are looking at that. This could help, especially going back to glyphosate-resistant palm amaranth with such a high amount of enzyme, I said, right? The RNAi technology helps us to actually target those plants that produce more enzyme or more messenger RNA. Basically, they stop making that protein so that the plants may not have enough enzyme. They will be still susceptible. That's something in the long run, though. Yeah, we are making baby steps in our lab. We are working in that area as well.
Do you mind taking a minute or two longer to kind of discuss what that process is for any of our listeners that aren't familiar with IPM technologies, and kind of break down a little bit further what you're talking about?
You mean in terms of RNAi?
Yes.
Yeah, sure. So RNAi, like I said, is not very new in pest management. What the technology itself, what it does is, what your listeners probably are aware that DNA makes RNA, RNA makes protein. That is the central dogma. We all are surviving on that theory. So when DNA makes RNA, subsequently if protein is not made, then That's it. The protein is not going to be in the cell. So RNAi, what it does is, when you have, instead of one strand of RNA, you end up having double-stranded RNA. That will not go further to make protein. So what we are inserting in the plant is to make a double-stranded RNA instead of a single-stranded RNA, so that the protein won't be produced. That is artificially done. Basically, we need a sequence of the gene of interest, and then you synthesize a double-stranded RNA and deliver it and see if you can reduce the protein produced. Thereby, not enough target available, so herbicide can still work in the resistant plants.
And in my understanding, this can be this can target very specific plants, very specific species. So therefore, it's not a broad, it's a very specific, which has a lot of us who are dealing with 3 and 4 way resistant weeds. There's a lot of excitement in being able to target kochia, as you mentioned, or to target.
Excellent point, Brian. Yes. See, we made a double-stranded RNA for EPSPS gene, which is the target site of glyphosate. We tested in palm amaranth. It didn't work. We did it for palm amaranth. We tested in water hemp. It didn't work. So even such a close relatives, not necessarily it will work. So it can be to our advantage, right? If you're targeting one species, just go for that technology.
That's fantastic.
It's time for our famous 3.
Well, as we wrap up, I've got three standard questions for you as we wrap up today in our conversation. And one is, what do you consider your go-to resource at work?
Yeah, very good question. There are a lot, but for me, this morning I had a meeting. Somebody asked me one question: what's the end goal of your research? I said, my goal is my research should reach the grower, ultimate user. For me, the stakeholder is growers. In that regard, if I need to know what are their problems, I go to extension specialists. I ask them constantly, have you heard any problems? Have you seen? Because we go to these national meetings and we learn, in other states, there is already PPO inhibitor resistant kochia in North Dakota they found, and in Canada I asked them, have you heard any Kansas growers complaining about lack of control? Those things, that's one thing. In terms of online resources, as everybody knows, they are available too. We go to, for my research, we have International Wheat Genomic Consortium, where we get all the gene sequence information. And also we have a very nice database where we can see how many herbicide species are resistant to glyphosate versus other herbicide, which is maintained by Wheat Science Society of America. Other than that, all journals pertain to Wheat Science program. I definitely access those too also for the information.
Well, I can tell work keeps you busy, but is there anything you enjoy in your free time?
Oh, yeah, I know. We all need some time to be away from work. Couple of things. I like gardening a lot. Obviously not in that time of the year, but I do grow a lot of houseplants as well. When weather gets better, I'm very passionate about my flower beds and also vegetable garden. And I learned Indian classical music back when I was growing up in India. I'm continuing that as well. Yeah, those are some of the things I like.
Sounds fantastic. And finally, where do people, if somebody wants to find out more about your work, what's the best place to go to find out about what you and your lab is doing?
Excellent. Yeah, no, we do have a Jugulam Lab website. They can access from Google, or when you go to our department website, there is a page for my profile as well, my research aspects that we do and what kind of teaching I do, what classes I teach. Those are major things where you get a lot of information. To access my publication, Google Scholar is a very good place to see latest publications from our lab. I do have a LinkedIn page and also Twitter page as well.
Fantastic. Well, Dr. Jugulam, thank you so much for joining us today. And folks, if you enjoyed this podcast, make sure you subscribe to the channel. That lets you get any updates and any new podcasts that we release. And make sure that if you learn something, share it with a friend, share it with a relative, and just help us continue this podcast. Leave a comment, give us some suggestions on who you'd like to see as a new guest. Mithila, thank you so much for taking the time to join us today.
Great to be with you, Brian. I thank you, our listeners, too, for listening to my interview, and also you all for the opportunity.